All right, everyone. So this week we're going to be talking about restriction enzymes and agarose gels. Hold on for the fun. So here's this thing. Do you remember this yet? Um, this week we're going to be talking about DNA and how to look at life at the DNA level. So we're mostly focused on up here. And if you remember our structure for DNA, we have this anti-parallel double helix structure. And just to remind you, some of the important things we need to think about for today's class is that the backbones are all held together with covalent bonds. Every bond we see here is covalent, which are very strong bonds that link this as one giant molecule. Whereas the base pairs are held together with these little dashed lines here that represents hydrogen bonds, which are relatively weak bonds. And this will come into play in a second. So at the end of last week, you should have had your plasmid DNA extracted and quantified, and you should get a little peak like this. And right, so I told you that if you have a absorbance over 260 nanometers, that means that you have DNA or RNA. And theoretically, we purified this plasmid DNA out of bacteria that had those plasmids, and we had the kanamycin in the media to make sure that only the bacteria that had the plasmids grew up. But if we see a peak like this, it tells us that we have DNA or RNA in there. It doesn't necessarily tell us exactly the DNA or RNA that we have. And so this week, what we're going to do is use something called restriction enzymes to cut our DNA so that we can see exactly the DNA that we have. Okay, so restriction enzymes are these enzymes. So enzymes are proteins. These and they cut DNA. Okay, and so this is a little chemical structure or a three-dimensional structure of restriction enzyme binding onto DNA. So this twist in the middle, that's our DNA helix, and these squiggles. We'll talk more about what these mean later on in the semester, but these things represent proteins. And so they're binding onto the DNA. And if you notice here, our DNA structure, our backbone is cut. And what restriction enzymes do is they recognize specific sequences of DNA and they cut the DNA backbone at those locations. Typically, those specific sequences that they see are palindromic. So if we look at this sequence down here, this is the restriction site. So the piece of DNA that gets cut by a restriction enzyme is called a restriction site. Um, so this enzyme EcoR1 cuts at the sequence GAATTC. And so this again is the 5' to 3' orientation in the front in the top here. So we would see that 5' phosphate here and that 3' OH down here. But if we look at the complement of this sequence, so the base pairing for that, we can see that we have the same thing in the opposite direction. So we have GAATTC read in the opposite direction. And so this is 5' to 3' this direction, and this is 5' to 3' in this direction. And this is what's known as a palindromic sequence. What you see this direction is the same as what you see in this direction. Um, and what happens when EcoR1 cuts at this GAATTC site is it cuts the backbone between the G and the A at both positions. Um, and so what that does is it leaves this single stranded. You can imagine that our DNA, right, it's double stranded. And when the backbone gets cut, that allows the bases to separate there in the middle. There's only four sets of hydrogen bonds holding these bases together. And that's relatively weak, so they kind of fall apart and the DNA can separate there, right? Because it's really these covalent bonds that are holding our DNA structure all together. And when we cut them, it allows, basically only the hydrogen bonds to hold it together, and that's not really enough. And so it separates. Okay. And if you think about, looking at a structure like this isn't exactly what would be considered a restriction site, but think about right here getting cut, and then like down here getting cut, right? The only thing holding that region together in the middle would be these 5 hydrogen bonds, and that would allow the DNA strands to separate there. And so if we look at our plasmid DNA, I mentioned this thing before, right? We have our promoter, our eukaryotic promoter that drives expression. We have our green fluorescent protein. We have our antibiotic resistant cassette and our origin of replication that I talked about before. But we also have this region called the multiple cloning site. And that multiple cloning site is specific for a reason because of all the restriction sites that are present in that particular region. One other thing I kind of hid last week were all these weird names, right? BSRG1, not one, EXPA1, AFL2, DRAW3, STU1, ECO109, APA-L1, ASE1. Those are all sites for restriction enzymes to cut. Anytime you see one labeled specifically in a plasmid like that, it means that it only cuts once. Um, but if we look closely at the multiple cloning site, so here's our multiple cloning site, and this next slide, we're going to zoom in on the actual DNA sequence of that multiple cloning site. So at the end of it, here's where our GFP starts. Okay, so you can see here's our GFP in this diagram. Our multiple cloning site is just upstream of that GFP. And so if we look here, all of these names, NHE1, ECO47, all the way down there to AGE1, those are restriction sites that are specifically in the multiple cloning site. And basically when people engineer a plasmid to be useful, they cram a bunch of restriction sites into one location so that we can use them. And we'll talk more about how we use these for cloning later on. But for today, just know that each of these restriction sites cuts once in the plasmid. And so one of the things that we're going to do is if we take this circular piece of DNA and we cut it at one location, that'll make that circular piece of DNA linearize, open up, and we'll be able to see a band of 4,700 bases, right? This plasmid that we said before is 4,700 bases long. So if we run this on a gel, which I'll talk about in a second, if we cut this, run it on a gel, we should be able to see a band at 4,700 bases long. But I'm also going to have you use another enzyme called DDE1. So if we look at our HindD3 site here, right, we see our HindD3 site here. This is the restriction site in the DNA. So it's AAGCTT, and you can see palindromic, it's AAGCTT. It cuts between the A and the A there. And so then, this leaves this 4-base sticky end hanging out there. You can see DDE1 recognizes only 5 bases, and one of those is an N. And you might say, wait, N isn't a base. Well, what that means is that any base can be at this position. So it doesn't actually recognize this position. It just needs to have this particular spacing. So it recognizes the CT and the AG here. And you can see this is palindromic, that direction, that direction. And so this is a much less selective restriction enzyme, which means that it's likely going to cut our plasmid in multiple locations. Anytime you allow some flexibility in the restriction site, it opens up places for the DNA to possibly get cut. And so I'm going to tell you, when you look at this HindD3, we know that this only cuts our plasmid once, which means we're just going to linearize our piece of DNA. But this DDE1 is going to cut our plasmid multiple times. And I'm going to ask you to do some stuff with that later on. So the restriction digest of this is going to look very different from this. So hopefully we'll be able to see that pretty clearly when we look at DNA gels. So how do we look at DNA? Well, so we use a molecule called Agarose. It's basically a sugar structure, long sugar chains. It's kind of a derivative of something called agar. So agar is a seaweed sugar chain polysaccharide. There's some more information here, but basically agar is a not super refined seaweed derivative. Agarose is very much more pure and allows for the things for DNA separation. Basically, if you have a high enough concentration of agar or agarose in a solution, it'll make something like a jello. Um, so agar is something that we use for making bacterial or microbial medias so that they're solid, so that we can streak out microorganisms on plates. Um, and it doesn't need to be very well refined in order to solidify. But to look at DNA, we need a really nice, pure, homogeneous matrix of our polysaccharide or sugar chain. And so agarose, you theoretically could use to make bacterial growth meat or microbial growth plates. But agarose is much more expensive. And so it's a huge waste of money, whereas agar is relatively cheap. So, that's what we use to grow things on. So just remember that for the future. Whenever we're making DNA gels, we're using agarose. And if we're making growth media, we're using agar. All right, so what happens here? So we have our little gel or our jello that we make up with our agarose. And that gel has little holes in it. We put the DNA down in those holes. Then what happens? Well, if we look at our DNA structure, one of the things I haven't noticed here or mentioned previously is phosphate, right? I mentioned phosphate, but one of the things about phosphate that I haven't mentioned is that every one of these phosphates has a negative charge on it. And that makes DNA overall have a very negative charge. And so what happens is if we take our matrix here, that's represented by these little balls inside this blue thing. Um, Essentially, it's like a liquid here that they are referring to the blue, and the dots are the agarose. If we take our DNA sample and we put it in a well in our DNA, and we apply an electric field, because that DNA is negatively charged, it's going to want to go towards the positive charge in the electric field. And so the DNA starts migrating through that matrix. Okay. And so if you give it enough time, it's going to move through more quickly or further. But DNA will separate out. This is called a chromatography technique. And anytime you do some sort of chromatography technique, you're going to be separating things out. And so, excuse me, our DNA will separate out based off of size, right? Because it's all similarly charged. And so the smaller pieces of DNA can move through this matrix of agarose quickly, more quickly than bigger pieces of DNA. The bigger pieces of DNA kind of get stuck up in that matrix. And so they move more slowly. And so what we do is we add a dye called aethydium bromide that intercalates in our DNA bases. And that allows us to see our DNA in a gel. It fluoresces when the aethydium bromide sits in the DNA bases. And so what we see is a little gel with our DNA in there. The way to remember the orientation is run to red. Okay, so red is the positive charge in an electrical system. And our gel boxes have black leads and red leads. The black leads are the negative, the red leads are positive, and so we always want the black leads to be over by our wells, and we want our red leads to go where we want our DNA to go. Okay, and this overall process is called gel electrophoresis. Okay, so electric movement, basically. This is just another way to kind of look at it. So you can see here, this is what our gels kind of look like. We have these little wells up at the top. We use pipettes. This isn't the best little pipette diagram, but we use the pipettes to load our DNA in the wells. And we apply an electric field down here, and you can see that the DNA moves through. One of the things you can see with this little diagram here is that there are these known sizes of DNA, and we call these a ladder. And you can see that these smaller pieces of DNA have migrated through the gel faster, and so they've come out further down here. The bigger pieces of DNA migrate slower. And so we can tell the approximate sizes of bands in our gels based off of how they compare to the ladder. Okay. And so, like this one right here, right? This band right here that we see in our gel between the 200 and 300 band. Um, so we can approximate that to be about 250 bases. This one right here is between our 300 and our 400. This will be 400 and 500 there. So this one's between the 300 and the 400. So this is probably around 350 bases. If we wanted to, we could theoretically generate a standard curve of migration distance, and then measure the migration distance and get more specific. But normally this gives us a pretty good idea of what our samples are by relative sizing, just kind of by eyeball. The one thing that you should know when you're looking at gels like this is, the migration distance tells us the size of the pieces of DNA that we're looking at, but the intensity tells us how much DNA we have, right? So this band in relation to this, there's much more of whatever this is than these two things, right? If we look at this is kind of lighter. It's probably in less, there's less of this DNA than there is of this one, which is less than that one, right? And so the intensity is the amount, the distance moved is the size. Okay, so those are the two things that you can tell from a gel like this: how much relative, how much you have, and how big your piece of DNA is. And the sizes are in base pairs, so the number of bases, so that rungs of the DNA ladder. Okay. Um, we have some different ladders that we use in the lab. We have 100 base pair ladder, so that goes from 100 bases to 3, 4, 5, 6, 7, 8, 9, 100, or 1,000, and some of them have a 1,500 size. One thing to note here, based off of the theoretical size of your DNA bands that you're looking for, you might want to change the concentration of agarose in your gel. Okay, so if we're looking at big pieces of DNA, like that 4,700 is a relatively big piece of DNA, we would want a lower concentration of agarose. If we want to do a small piece of DNA, like this 100 or 200 bases, we use a higher concentration of agarose. And so this one is showing on a 2% agarose gel. 2% is sort of the maximum. And generally like 0.7, 0.8% is the lowest that we wind up using in this lab. So somewhere between 0.7 and 2%, we alter our agarose concentration. That lets us see different sizes of DNA easier. So this ladder is called lambda phage, bacteriophage lambda. Basically, its genome has been extracted and cut with the restriction enzyme HindD3, and it creates this banding pattern, right? And so this one we can see bigger stuff, right? So there is a 564 base band right there, but it's a little lighter. You can see here we're going down to 100. Mostly this one is useful for things from around 2,000 up to 23,000 bases. And you can see that this one is in a 1% gel, right? This one was a 2% gel. We can see these smaller bands better. This one is a 1% gel. We can make out some of these bigger bands easier. There are ladders that we have. This one's called size standard, that basically can combine two different sets. So if you really don't know what size piece of DNA you have, you could use something like size standard, where you get everything down from 72 bases up to 23,000 bases. So it's a way to see a lot of different stuff. And again, this one would probably 1.5 to 1.8% gel, somewhere in that range. And so when we think about the structures of DNA, I've kind of been talking about it as these linear molecules, But in reality, DNA has to be packaged. It's a really long molecule. So if we take the example of E. coli here, right? We said before that E. coli have 4.6 million bases of DNA. If we do some basic napkin math here, 4.6 million bases times 3.4 nanometers per base pair, right? So this will tell us how many nanometers our piece of DNA is. And so it's 15 million plus nanometers long. If we convert that to micrometers, right, we're just removing decimal places as we go from nanometers to micrometers, 15,000 micrometers. Again, we're moving 3 decimal places to go up to millimeters, 15.64 millimeters. If you think about millimeters, that's actually 1.5 centimeters, right? 1.5 centimeters is actually something that you can see. E. coli are only 2 micrometers long. Essentially, the DNA that fits inside of an E. coli is 7,820 times longer than the bacterium in which it's crammed into. And so bacteria use a technique called supercoiling to essentially twist the DNA up, and that allows it to get condensed down into these little knot structures. Eukaryotes use things called histones, which are shown here. All right. So as she mentioned in that video, eukaryotes use these things called histones to wrap our DNA up into these tighter structures. And as she mentioned, I mentioned before that the human genome is 3.2 billion bases long. Essentially, every cell in our body has 6 feet of DNA in it. And so that's pretty wild. And so thinking about something that's that long getting condensed down into a tiny little thing like a nucleus, that shows you how packaging has to happen. And so if we don't wants us to watch this again. And so if we think about our plasmid DNAs, they need to be packaged as well, right? That supercoiling that the bacteria does. And so if we were to take our plasmid DNA and run it directly on a gel, it's going to run weird because it's going to be all supercoiled up. And so that's why we need to cut our plasmid DNA with the restriction enzymes. What they do is, the plasmid's all coiled up. We cut it with a restriction enzyme and that loosens it, turns it linear, right? And so we'll be able to tell the difference whether our DNA is cut or not on the agarose gels. So when you run your gel, our new gel apparatus doesn't have to worry about focusing, it auto does it, but you want to think about how your gels look. And you never want to turn in a gel that looks like this, really. Um, one of the things is that there's no labels on it. You always want to label what goes into each well. Okay. And so you can just use like Google Slides or PowerPoint to label up your gel and submit it that way. And so basically you want to say, okay, this lane has the whatever ladder, and you want to say what kind of ladder it has. This one has our DNA that's cut with X. This one has our DNA that's cut with Y. This one has some DNA that's cut with whatever. Um, and so you want to label what goes in each lane. Don't ever just submit an empty gel like this. Here's one that's labeled well, right? You can see this gel has run a lot nicer. Here's our wells up here. The red would be over here. The black would be over here. And so it goes this way. You can see we have a 1 kilobase ladder that's been used in this particular one, which is one I haven't talked about, but there's lots of different ladders. This is telling you what's in each lane, and you can see that you can get a relative idea of, the intensity and the size for each of the bands. And so this is the kind of thing that you should be submitting when I ask you to submit a picture of a gel. All right, so this week, what you're going to do is you're going to take that plasmid DNA that you extracted last week, and you're going to cut 250 nanograms of it with the restriction enzyme HIND3, okay? Then you're going to cut 750 nanograms of it with the restriction enzyme DDE1. Okay, so you're going to set up these two restriction digests. Maybe it takes you half an hour to set up. Basically, you can let restriction digest go overnight, or you can come back in about a half an hour. It all depends on what you want your schedule to look like. So if you just want to spend a half hour setting this up and then letting it go overnight, just sign up for a half hour time slot, set this up, come back the next day. The next day, what I want you to do is take all of this DNA, and 100 nanograms of undigested plasmid DNA. So you're going to have your ladder and then 3 lanes. So your ladder, this one, and then this one, and then this one, on a 1.5% agarose gel to determine the size of the bands. So this is about 2 hours to make and run the gel. Okay, so you're going to have to dissolve the agarose. The protocols are up on Moodle. Heat it up, and then physically run the gel with the run the gel electrophoresis. Okay. Theoretically, you could make your gel and have it starting to cool and solidify during this 30-minute incubation. So if you wanted to do it all in one session, once you set up your restriction digest, there's no reason to wait this whole 30 minutes. Start making your gel now, let it solidify and cool. And then, it'll be ready right after that 30 minutes to run your gel. So it's a way to sort of optimize your time, and thinking about time optimization and all these different protocols is something that's going to come a little bit more naturally the more experience you have doing it. But if you want to take more time and do them separately, that's totally cool too. So then you're going to take your gel, take a picture of it in our gel doc, and then label it up, and then post it to Moodle up on our class forum. All right, so have a good week.